So wait, you know you transport the public with public transport, right?
Yeah.
So how do you transport proteins?
Cannons.
Ah, so protein transport.
Yep. So eukaryotic cells are magic.
That is pretty much the video.
Yep, that sums up the rest of your course.
Yeah. Biology's magic. We don't know how it happens, but it's great.
And in case you want to know how it's magic, that's what the rest of the video is about.
Yeah. So we mentioned before that the nucleus and nucleus have ribosomes and stuff, and things move from pores and all that jazz. But now I'm going to say what actually happened in a bit more detail.
So here's, this goes on. This is the whole of the nucleus here. This is the membrane. There's little pores in it.
Yeah.
Inside is lots of ribosomes, which I'm going to express as dots, and then there'd be a nucleolus, which I'm just going to put here. It's sort of a dense lot of ribosomes.
Yeah. Okay.
And that has the raw materials for making proteins. But I've noticed a problem.
What's that?
A protein has got to be exactly this big. And it's not going to get through here, is it? So if we make a protein inside the nucleus...
Won't for now, because all of our drawings are drawn exactly to a very good scale.
They are indeed.
They are indeed.
Cracker scale. So a protein would be too big if it was formed in here to get out of the nucleus.
It's kind of like building your wardrobe in IKEA. You can't get it back home.
Unless you have a van or a lorry.
So then what you have to do is bring your flat pack to your house.
And then build it.
And then build it, which is basically what happens here.
It is indeed. Nice way of thinking about it. Always think of IKEA.
I love IKEA, especially their meatballs.
Exactly. So, it has the raw materials for making a protein, and it sends them out the pores because they can fit the raw materials, the stuff. And that goes out
the pores. What's that going to then?
Well, that's the question. What does it hit first? And close to the nucleus is rough endoplasmic reticulum.
Rare.
So sort of it goes through a channel, and then it goes...
Yeah, into a weird, like, maze shape.
Yeah, I'm not going to do it too big. In your biology book, it might be a little bit bigger than this. It's actually drawn. But it goes in there.
When it goes through, it folds into different shapes, assuming more of a protein type shape, of course. Yeah, as Wayne remembered. It's rough, so it's covered in ribosomes.
Yeah, absolutely. Because the smooth endoplasmic reticulum is for lipids and stuff, which we don't really do much of in this course.
So that's what ribosomes here. There are ribosomes everywhere else about, but these things are doing stuff for inside the cell. These ribosomes, this stuff is for extracellular, outside the cell.
So, this protein is for outside the cell. So it's going through here, it goes through the maze, it goes this way and goes, oh, wait a minute.
Gets a bit lost, goes through, comes out, looks quite different, but how does it come out then?
Well, imagine this is like a goo. It's not a goo, imagine it like a goo.
Right.
You try and push something out, something's going to be inside a little goo ball.
Yeah, it's kind of how it works.
And
so this bit in stages.
Yeah, so it's sort of in this bit and then it gets pinched off inside this thing, and we call it a vesicle.
Yeah.
This ball with the stuff in it is called a vesicle. But it's not done yet, it's not completely processed yet. It looks a lot more like a protein now. It's not just raw materials, but it's got another step before it can be a protein, the correct protein.
Yeah.
So what Trev's drawn here, it looks a bit like a spider in the corner, and I love that. It's a lovely spider.
Spiders are Golgi Apparatus, that's how that works. Loosely like that.
So this is a Golgi Apparatus, and this is the second part that it goes to.
And
it's pinched in, just like it was pinched out.
These two things have names, which you think you need to know as well.
Yeah.
So here, this will be called endocytosis.
Yeah, there's no easy way to remember that.
No, but this one is called
exocytosis.
That's the other way around, isn't it?
No, it's not. Exocytosis is when it's going into something, it's exiting. So it's exiting the vesicle and going in. And then it goes through the Golgi apparatus and it's pinched off in like a ready-made ball at the other end. It's ready-made balls.
And this ball...
It's like ready crack, but ready-made.
Yeah. And that's after it's got a few further processing, so it's now more perfect how it wants to be.
So it's trimmed to the correct size and it's proper three-dimensional stuff going on now.
Now we're at the edge of the cell here.
And again it does exocytosis.
Because it's exiting.
It's pretty similar stuff.
And it's sort of gone, all coming out here, and this is fused back to how it used to be.
Yeah. And that's how raw materials make proteins and stuff.
Yeah, and at least move around proteins.
So this is eukaryotic. You know that Golgi and the rough endoplasmic reticulum is not in prokaryotic. Most of this course does focus on the eukaryotic, so think about that.
Let's focus on this exam, and that's what we're going to focus on for the next few videos as well.
So let's crack on.
Alright.